IFB microscopy and Zeil-Nielsen staining. Dark screen slowly revealing a glowing microscope in a laboratory. Dramatic lighting with floating dust particles. Did you know that a tiny bacterium under a microscope can reveal one of the world's deadliest diseases? Close-up animation of red TB bacteria moving across the screen. Tuberculosis continues to affect millions of people worldwide. Ethiopian laboratory technician preparing slides in a realistic medical laboratory. Every day, laboratories use AFB microscopy to help diagnose TB quickly. Microscope lens focusing dramatically onto a stained slide. Today, we explore the Zeil-Nielsen staining technique step by step. Host standing beside a laboratory bench with microscope and staining reagents visible. Welcome to the channel. Let's learn how scientists identify acid-fast bacteria. On-screen title animation, AFB microscopy, and Zeil-Nielsen staining. This method remains one of the most important tools in tuberculosis diagnosis. Graphic showing lungs infected with tuberculosis. The bacteria responsible for TB are called acid-fast bacilli. Scientific animation showing the waxy bacterial cell wall. Their waxy cell wall makes them difficult to stain with ordinary dyes. Split-screen comparison between ordinary bacteria and acid-fast bacteria. That is why a special staining method is required. Red rod-shaped bacteria appearing under microscope animation. When stained correctly, acid-fast bacteria appear bright red. Blue background cells appearing beside red bacilli. The surrounding cells and background appear blue. Laboratory bottles labeled Carbol Fuchsin, acid alcohol, and methylene blue. The Zeil-Nielsen method uses 3 important reagents. Technician pouring Carbol Fuchsin onto a slide. First, Carbol Fuchsin stains the bacteria red. Gentle heating of the slide over flame with visible steam. Heat helps the stain penetrate the waxy bacterial wall. Acid alcohol washing stain away from non-acid fast bacteria animation. Acid alcohol removes the stain from ordinary bacteria. Red bacteria remaining visible after decolorization, but acid-fast organisms retain the red color. Methylene blue added onto the slide. Finally, methylene blue provides background contrast. Laboratory technician preparing sputum smear on slide. The procedure begins with preparing a thin smear from the specimen. Slide air drying beside laboratory equipment. The smear is air-dried before heat fixation. Flame heat fixing the slide carefully. Heat fixation helps attach the specimen firmly to the slide. Carbol Fuchsin covering the smear completely. Next, the slide is flooded with Carbol Fuchsin stain. Steam rising during gentle heating process. Gentle heating allows deeper stain penetration. Water rinsing stain from the slide. The slide is then rinsed with clean water. Acid alcohol applied carefully to the smear. Acid alcohol is added to decolorize non-acid fast organisms. Counterstaining with methylene blue. Afterward, methylene blue is used as the counterstain. Microscope oil immersion lens moving into focus. The slide is examined using the oil immersion objective lens. Microscopic view showing bright red rods on blue background. Red rod-shaped bacteria indicate a positive AFB result. Healthcare workers treating TB patients in clinic. IFB microscopy helps healthcare workers diagnose TB rapidly. Rural health laboratory in Ethiopia with simple equipment. The method is affordable and useful in resource-limited settings. Modern molecular testing equipment beside traditional microscopy setup. Although newer technologies exist, microscopy remains widely used. Laboratory worker wearing gloves, mask, and protective coat. Safety precautions are essential when handling infectious samples. Proper disposal of contaminated laboratory materials. Proper disinfection and waste disposal help prevent infection spread. Montage of microscope work, TB awareness posters, and laboratory teamwork. Zeil-Nielsen staining continues to play a major role in public health. Host speaking directly to camera with microscope beside them. Understanding this technique is important for students and healthcare professionals alike. Closing microscope shot slowly fading into channel logo. Thank you for watching. Don't forget to like, share, and subscribe for more laboratory science videos.